All right, so I'm Dr. Cyril Pernet, so I'm the chair today with Pierre as well, who's there at the front. So in this first talk, talking about reproducibility, the questions that I wanted to address, so you probably all have read papers about it, one of the most famous being this reproducibility paper in psychology. And so the question is, does it apply to brain imaging? And a slide I borrowed from Dorothy Bishop, which highlights issues in biomedical sciences. So you will be familiar with this paper from PLOS, which highlighted the issue. That's pretty much where it all started and why there's too many false positives in science in general. And then there's many others. So this paper about the reproducibility in psychology, which was actually a paper in Science. That's just the highlight from Nature. And some others highlighted there. And so just before we dive into the other lectures, introduce the concepts and the differences between reproducibility, repeatability, and replicability. So all of them are pretty much used, interchangeably. And so I took this definition here from a paper published in Nature Method, repeatability being simply repeat what you've done, right? Which means under the same conditions, can you rerun your own same analysis a second time and get exactly the same results? So these highlight issues that you need to probably write code rather than just click on GUIs. You need to document, and also issues of provenance. For instance, which version of a given software you use. The reproduction is the idea of recomputing the same things. It's essentially basically an issue of your research being transparent for someone else. So again, in this case, if you want anybody else to be able to reproduce what you've done, you might want to share your data so that they can run the same analysis on your data. You probably want to share your code because there's many different ways you can realign, many different ways you can normalize, many different ways you can do various statistical models. And the replicability is the idea of achieving consistent results. And so in this case, somebody would collect a new sample, possibly use your code, and then we would simply want to see whether or not we can replicate the results. And that comes into issues of design and issues of power, right? If you want to replicate somebody's studies, you need to make sure you have enough participants so that you have enough statistical power to get chances to find an effect if the effect is there. Two more, a little bit more in the definitions. So reproducibility has to do with the credibility of the result, whereas the replicability, the fact that we can find the same thing a second time with a new data set, possibly with a different analysis, has to do with increasing your evidential weight. And so again, you will probably in different papers, you'll see things about method reproducibility. So that's to do with basically sharing your data and code, so we can make sure that people can do the same thing twice. Whereas the result, which is basically the replicability, we want the same results. And inferential reproducibility refers to conclusions vis-à-vis hypotheses of interest. And that's basically cumulative sciences. So it's like if we all share the data, we can accumulate enough information and we can weight that information whether or not an effect is real or not. Just to highlight that idea of reproduction as being a continuum rather than an absolute thing. And so what we're going to be talking in the next few sessions is going to be different aspects along that continuum. We're not saying everybody's got to do everything the same way. It's like, depending on what you have, what your ethics committee says, and so on, there's various ways you can work toward being reproducible. And so you can start by make sure everybody can do that first step, which is everybody should be able to repeat their own analysis, right? And so it means if you are in the lab, whether you're a student or you're a PI, whoever does the analysis, to make sure it's well documented so that anybody else who comes up will be able to redo it for yourself, but anybody else in the lab. And so that's the second step, which is reproductions of someone else's analysis, right? So to start with, you should be able to do it in your own lab, and then ideally, we should be able to do it. I should be able to come to you, you give me your code, and I should be able to redo the same thing as what you've done. And then you've got other aspects, which is the direct reproduction. So same experiment, but new sample. Then you've got the systematic reproduction. And then you've got the last step, which is conceptual reproduction, this idea that we can replicate some finding, providing that the concept behind an experiment exists. And so what can we expect from that? I quite like, there's not a very famous paper from United, but I like this idea, is that you've got variability in biological systems, whether that means we don't expect that you're going to find exactly the same if you do the same experiment as me, right? Because you've got variability in your samples. The issue really that we want to focus on is the fact that we can't obtain the same results overall, and those issues relate to experimental design and power. They're designed to cognitive bias, which has to do with the way you do things, so we can deal with that. And it has to do with the statistical analysis, which often are not as good as they could be. And so there was this survey done by Nature in 2016, and basically they were asking a range of researchers, not just from brain science or neuroscience, but life science in general, and they were asking whether they think, do you think there is a reproducibility crisis in your community? And so most people would agree that there is at least some problem. You know, we can still see some outliers there who think that everything is superb in their field and there's no problem at all. But most people agree that there is a problem. And then when you break it down, they're looking at, well, what kind of problems we have. And so what was interesting in this one is that we've got a few of them that we can address. And so I can't really read from here, sorry. What's the first one? Yeah, I can't read from here. It's too far away from me. But we've got a few, I'll get in the next slides anyway. Yeah. And so selective reporting, the first one. And so for that, we have NeuroVault, and Chris is going to be talking and presenting that tool. Then we've got these issues of power. So now everything is solved, but we've got fMRI power and neuro power, and Janet's going to presenting how you can do some power analysis for fMRI. Then another big issue in that survey was people talking about not being able to repeat enough or no code sharing. And so Pierre's going to talk about containers, pipelines, and various ways you can make your code easier for other people to reuse. Issues of designs, and so Pierre's going to talk about pre-registration, which is one way to make sure that your designs are optimized to answer the questions that you have. And then we've got data sharing. And for that, basically, INCF has been working on beads, and Cameron will be presenting, which is a specific format in which you want to share your data. It's very easy, you'll see, if you never looked at it. And we'll have sessions as well. I have to say that we also have sessions on Tuesday, in the open science room. So today what happened is that we'll have all of this from a theoretical perspective, and then during the week you can come and see, and we'll have more talks and demos and so on, a bit more practical. And so I'm just highlighting, if you do EEG and MEG, there is a bit of a lack of tools, and because all of them are very well designed for MRI, possibly PET, but there's actually not much going on for MEG. So if you do MEG, then there's a room for you to create new tools. So I'm just going to finish the talk talking about cognitive biases, which is something that we don't really have. The tools are designed basically to fight these biases. And so the typical scientific method works this way, and Pierre is going to show the same diagram later on today. And so we start with an hypothesis, we go into the study design, we collect the data, analyze them, and get some interpretation, possibly publish it. And so the first biases that we have is this IKEA effect. And so that's typically marketing issues. But that's the idea that, you know, as a consumer, you place a high value on stuff that you build yourself. And so if you think about it, in neuroimaging, we do tend to code a fair bit. And so clearly, that means that Your experiment that you designed yourself is necessarily the best one because you designed it. And the code that you're going to use is necessarily the best code, right? Your support vector machines that you coded yourself is necessarily the best support vector machine because you built it yourself. And so we've got a bit this problem in the community that we all tend to do things ourselves, and we always think that what we do ourselves is always the best, which is not necessarily true, right? Because that IKEA stuff that you have in your house is not necessarily the best furniture you have in your house, but you build it yourself, so you like it. That's the same idea. The other bias is, it's got various names. One of them is being called the Texas Short Shooter effect. Is the idea that you shoot and then you put the target afterward. So necessarily you're going to always shoot right in the middle, right? Because you put the target after. That means that basically what we do is we try many analyses, techniques, until you find one which starts to show the results that you expect. So instead of designing in advance, saying this is what we're going to do, and the results show up or not, what we do is we try many of them. At some point, we find the results and say, that was the one I wanted. That's the one I wanted from the beginning. It's just, I tried something else first. And that leads to p-hacking. Because of course now, if you do many times the same, you try many different analyses on the same data set, at some point by chance, you're going to get a significant result. That doesn't mean it's the right analysis. It could simply be just chance, right? Hence the issue of p-hacking. And so again, I put pre-registration there because since in pre-registration, Pia will talk about it, but the idea is that you would define in advance how you can analyze the data, means that would prevent you from trying some things that you didn't think of just because the results are not significant. The other one is confirmation bias, and so this one basically is everywhere. And that's the idea that you're going to carefully debug your analysis and debunking the data that counter your favored hypothesis. But then you're going to just not really care about evidence which goes against your hypothesis. And so a very simple solution against confirmation bias is data blinding, right? People who are involved in clinical trials know about this. It's something which is very common. We do use a double-blind clinical trial. You don't want to know about which one is your patients, which one is not is your control group, and you do your statistical analysis blinded. But that's not typically what we see in neuroimaging. It's the same people who do the experiment and do the data analysis. And that means this, with this idea of using multiple different ways of analyzing the data, leads to basically simply do an analysis which leads to what you expect. And so you just confirm rather than trying to infirm. And so the question that I put there for you is, how do you know that the analysis you've done is correct? Well, because it gives the results that I expect. Right? So you do a given analysis for fMRI or MEG, you find the results, that's what I expected. So you know it's true. Your code works because it gives the right results. That's confirmation bias. Did you ever correct the code which gives you the right result? Right? You correct your code because it doesn't spit out the good result. So you say, there's a bug. If it spits out the right result, there's no bug. It's necessarily good. But how do you know it is good? It could be a bug that gives you the right results. At least the result you expect. And so the last bias that exists and plague the scientific method is the hindsight bias. And this is the inclination after an event has occurred to use the event as having been predicted. And so that basically leads to, in conjunction with publication bias, but that leads to hacking and harking. And so that's the fact that basically you come up with an hypothesis after you've seen the results. And then there's also sharking. So I quite like, so that's Russ Poldrack who came up with it this year in 2016. So instead of harking, which is hypotheses after results are known, the sharking in selecting hypothesis areas after results are known. Right? That's I'll just do a contrast. I find some pattern somewhere in the brain, but it's not significant. And then I come up with some a priori regions of interest right in the spot where it's a little bit higher, there's a little bit more activation, so that I don't need to do a full brain comparison. I can do ROI. But I've seen the result in advance, so my ROI is biased, right? And so that's sharking. And again, this is the same as harking, right? You come up with the idea, oh yeah, but I've seen in the literature people using that region. Except that you came up with this idea after seeing the results, whereas you should really have this idea before. Because if we search enough the literature, we're going to find regions of interest for whatever we need. And there's enough paper out there to justify a posteriori. And so that relates to this idea of also of circularity analysis and independent regions of interest, which is you can't really use the same data to define your ROI and get estimates from it. And so we've got all these biases, and a lot of the tools that we're going to see today, pre-registration, statistical power, pipelines, and data sharing, are helping to fight these biases. Another aspect that is not really touched because we don't really have tools for it, but it's good to be aware, is these issues of analytical complexity. And so there is this paper now, relatively old, 2012, if I remember, from Joshua Scarp. And so starting from the paper from Analysis in PLOS, the idea that we have a lot of false positive, depending on mainly relying on differences in methods. He's been analyzing a given data set, basically just using the standard FSL, SPM, AFNI, and then combining every possible pipelines you can do using these and whatever parameters you can change within the different algorithms. So we can see here he's got 6,912 unique analysis pipelines once you combine all three of them plus whatever parameter sets that you can change. What's interesting in it is that you would get, so on the left side is the mean activation across the different pipeline, whereas on the right is the analytical range, so how much variations we get. And so you can see that even in some regions where on average you've got some effect, like on the right-hand side of the brain, this very same region can also show very large variability. So even though you would find it a lot, the actual values that you measure in there can be very different. You also have other regions which can show up highly variable even though the mean is not consistent. You also looked at the stability of the results in terms of peak of activations, and clearly we don't really have any overlap between the different methods. And so what's interesting thinking about this issue of stability and variability is that we have many tools for which we can analyze the data. There's many different ways we can do it, and this leads to variation from study to study. And so simply saying in a paper that you use realignment and you use FSL is not necessarily good enough, because some of the parameters of the default settings, or at least you would need to say that you use the default setting and which version it is to make sure that we can redo the same. So it's very difficult to find the exact same results. And that's pretty much what that paper shows, is that even though it's the same person who do the analysis, simply combining the different softwares and using different algorithms, you get different peaks, you get different values. And so even though the regions show some stable results, this stability of the result is only for the very strong effect. And effect size and precise locations vary a lot. So in that paper, vary up to 8 Z units, which is absolutely huge. And this leads to very large differences once you start to threshold, right? Because if you've got, in terms of Z values, 8 Z values difference, and you decide to threshold at a given value, then of course now you're going to have very, very large variations. And so the patterns were the same, but the results were different. And so what that means, it means for you, you must declare all analysis performed, right? If we do a different type of modeling using different basis sets in your GLM, for instance, you might want to say that you did use different sets, but only results come out for that particular set. We need to share the map, because if you share the map, we can compare effect size. So it's very likely that, for instance, you have a blob, I don't find the blob, but if I look at the actual raw map, I have an activation at the same location. It's just, in my case, it's significant because it's above the blob and it's not for you. That doesn't mean that we don't have both activation in the same region, right? It's just either we talk about binary outcome, do we have an activation of the same place, or we are talking about, well, can we see an increase of activation in that region, which is not quite the same. And so if you share your map, then it becomes very easy for me to say, well, I can see that you didn't find a significant effect, but you did find an increase of activation in the same region as I do. Which somehow, I'm pretty sure you would agree that it's a stronger result by saying, well, we both see activation in that region, even though they are not significant individually, than I find something and you don't, right? And so to finish, the COBIDAS report is something that the OHBM came up with. So COBIDAS stands for the Committee on Best Practice and Data Analysis and Sharing. That was a committee put in place to specific to MRI and to look at the best practices for sharing and data analysis. And so if you go on the OHBM website, COBIDAS report, PDF, it's a very, very long document. But it's useful for you as an author because it allows you to know what are the different steps that you need to put in your paper and what are the different things that you can do. And it's also very useful as a reviewer. And so I would recommend using the Appendix D. Basically, it's a tick box exercise. And so if you review a paper, you can say, well, the best practice in the community are, and then you just basically go through your Annex D. It's pretty much the same when you prepare your paper. You go through Annex D for MRI and you check that, you do comply with most of the boxes in there, such as describing various parameters for your data analysis. And so what are the tips which come out from the COBIDAS? Provide enough information to the reader. That would allow to understand potential bias in your study. So what means demographic, clinical information, exclusion, inclusion, attrition, these kind of things that we don't tend to report even for normal healthy subjects. But this could matter. That's something very typical in clinical trials. Again, not something that we do a lot for healthy subjects. What is your pre-processing pipeline? And so because of this problem in journals where often we are constrained by the number of words, we end up writing things such as pre-processing, standard FSL or SPM pipeline. And that's it. That doesn't really describe very well what you've done, right? And so we need to justify, what's very important as well is to justify deviation from what the community tends to do. So if there is a typical way that people do things and then you do things differently, somehow you have to come up with why did you do it that way and why it matters. Then you've got what are the physiological assumptions in your data analysis. Again, things such as reporting the HRF models that you use. What is the rationale for multiple comparison correction? So we all know that we need to do a comparison for familiar-wise error rate. Question being, why did you choose one technique rather than another? And basically, it comes down to one very big tip, which is what the COBIDAS is really about, is to be transparent. We know no experiment is perfect. Certainly, my experiments are not perfect, nor are my papers, but what matters is trying to be as transparent as possible, because it means you can try to redo the same thing as I did because I share whatever I've been doing. Right? It's not a question of being the best study, it's a matter of being, I'm transparent so that you can see what I've done and reproduce it. And then we can learn from each other's mistake, and we can accumulate science and information by sharing the data and the results. So not everything is solved. But I think compared to some other fields, the neuroimaging community is actually not that bad. There's a lot of work which has been done, especially for MRI. And so this year, tonight will be announced the OHBM Reproducibility Award. And when we looked at these studies, what we see is that we do have now studies where we try to reproduce the design using the same stimuli, but it's because people share their experiment and share their stimuli. We also have a bunch of studies which try to replicate analysis on multiple cohorts, and all of this is thanks to data sharing, right? Because we've got these big databases, so now we have multiple studies which use the same databases, and so we can see if we have the same results, and using different techniques, what different results that we obtain. And so I think the community has been very good for this. And you will see some of these tools today. And then you also have like the true replication, right? Let's redo the whole thing. But what we didn't see in the applications today are lack of power analysis. So it means that with the increase in data sharing, the next step now is power analysis. If you want to replicate a study, there's enough data out there for you to pick up some maps or pick up raw data and conduct power analysis to design your next experiment. And that will reduce your number of false positives and negatives. And again, lack of pre-registrations and protocol papers. So Pia will talk about this today, and hopefully she will convince you why this is something that you need to do if you want to replicate. And that's it. So the next speaker will be Janet, and then Pia. Then we'll have a break, and then we can have a little discussion. Thank you.